Hello and welcome back to EaseGenetics, where we make genetics easy. I'm Dr. Kaveh Zargeri, and with 28 years of experience teaching genetics, I'm here to solve another typical problem regarding genetic linkage. Another key problem-solving skill in the genetic linkage is determining the middle gene with no D.C.O or double crossing over types among offsprings. So let's see how we can solve this tricky problem and identify the middle gene. First, let's see why we sometimes don't see double crossover types among offspring. The first reason could be that, as I mentioned in my video Linkage 201, 3-point gene mapping, the distance between the three genes is less than 5 map units. Due to interference, double crossover types will never appear among the offspring. The second reason could be related to the number of offspring, meaning that even though the total distance between the three genes is more than 5 map units, due to high interference, a large number of offspring is needed for the chance of observing double crossover types. In this video, I'll explain this further while solving an example. A sample problem is as follows. By test crossing a tree hybrid, the following offspring were obtained. As you see, the genotypes of the progeny are provided along with their respective numbers, which essentially simplifies our task by skipping the step of converting phenotypes into genotypes. At the end, there are two questions. I draw the genetic map, and assuming an 80% interference, how many offspring is needed to see the least number of D.C.O types among the progeny? The offspring are listed in a scattered manner. To simplify solving the problem, it is better to arrange them as explained in the Linkage 201 video. There we called it reverse pairs. Here, the offspring are organized into three groups of reverse pairs. The first pair corresponds to the parental type due to the highest numbers. Pairs 2 and 3 correspond to single crossover types. Now we have reached the most critical step in solving the problem, determining the middle gene. Here, we do not have double crossover types among the offspring to compare with the parental types and identify the middle gene. However, what we can do is hypothesize their genotypes. In other words, if a double crossover type offspring were to be formed, what genotype could it have had? What we do is select one genotype from each reverse pair as a representative of the group. At this stage, there is no need to write the genotypes of the chromosome inherited from the test cross parent, which is completely recessive. Only the chromosome inherited from the tree hybrid parent needs to be written. To simplify and avoid mistakes, write the four genotypes as follows. 1. Write all alleles as recessive. 2. Change only the first allele to dominant. 3. Change only the second allele to dominant. And 4. Change only the third allele to dominant. Now you have one representative from each of the four groups of reverse pairs. On the right side, you can see the reverse paired genotypes of the following offspring chromosomes as mentioned in the problem. Let's see which is missing. Look at chromosome 1, and you see it exists in pair 2. Look at chromosome 2, and you see it exists in pair 3. Chromosome 3 does not exist in any pair, while chromosome 4 exists in pair 1. Therefore, chromosome 3 is one of the two chromosomes resulting from a double crossover whose count along with its reciprocal is 0 in this problem. As I mentioned in my video Linkage 201, comparing one of the two double crossover types with the parental type is sufficient to identify the middle gene. Here I have written the same chromosome shown in the previous slide, and above it I have written the chromosomes of the parental types. By comparing the double crossover type with the parental type, it's clear that it is more similar to chromosome 2 as genes B and D are the same, while gene A has changed. As a result, gene A is the middle gene. From now on, in the parents and single crossovers, genes A will be placed in the middle. By arranging the order of the genes, we identify the single crossover in region 1. By tracing chromosome 1 in the parental chromosomes, shown in green, and chromosome 2, shown in purple, we see that the single crossover in region 1 involves a crossover between genes A and D. For region 2, by tracing chromosome 1 in the parental chromosomes, shown in purple, and chromosome 2, shown in green, the single crossover in region 2 involves a crossover between genes B and A. To calculate the distance between genes A and D, we sum the offspring resulting from the single crossover between these two genes, which is 20 plus 20, and add the double crossover types, which in this case is 0. This total is divided by the total number of offspring, which is 500, and finally multiplied by 100, resulting in 8%. This means the distance between the two genes is 8 map units or centimorgans. Similarly, for the single crossover in region 2, which involves genes B and A, we calculate and obtain a crossover rate of 4%, corresponding to a distance of 4 map units or centimorgans. To answer part I of the problem, we draw a hypothetical chromosome segment and place the genes on it in the correct order, showing the distances between them. To answer the second question II, we must consider that to observe the minimum number of double crossovers means that there must be one of each type, so their total will be 2. The question states that the interference is 80%, so the coefficient of coincidence is equal to 1 minus the interference, or 20%, or 0 and 2/10. As I mentioned in my video, Linkage 201, 3-point gene mapping, the coefficient of coincidence is the ratio of observed double crossovers to expected double crossovers. The observed double crossovers are equal to the number of offspring resulting from double crossovers seen in the offspring divided by the total number of offspring, which here is our unknown, and the expected double crossovers are equal to the single crossover frequency in each region. By substituting the numbers into the formula and solving for the unknown x, we find that the minimum number of offspring required to observe at least one offspring of each type resulting from a double crossover is 3,125. You can see that with these relatively short genetic distances totaling 12 map units, and considering that within this range there is a 5 map unit region where double crossovers cannot occur and only an 8 map unit region where double crossovers are possible remains, and given the high level of interference, 500 offspring is a very small number to observe a double crossover event. However, it's important to note that since we're talking about probabilities, we can't say that it's impossible to observe a double crossover with fewer than 3,125 offspring. Someone might be very lucky and observe a double crossover even with fewer than 500 offspring. Since we're dealing with probabilities, we can just consider it a lucky chance. Thanks for watching this video. I hope it was useful to you. If you enjoyed watching this video, please give it a thumbs up. If you want to watch more videos, please press the subscribe button. Feel free to share your thoughts, questions, or suggestions in the comments below. I hope to see you in the next video. Goodbye.